So good morning. Can I make sure I can be seen and heard? Very good, thank you. All right. All right, we'll get started in a few seconds here. All right, so hope everybody had a pleasant weekend and woke up, weren't too surprised waking up to winter this morning. I know normally we'd have places to get to, but maybe wasn't the case for me, but in any event, here we are. So we'll go through the quiz real quick and note that exam 3, this is not a quiz, this is a big exam, 75-point exam. Well, will be available. I'm going to give you all weekend to do it. It's an open note. I put 11 a.m. I guess I forgot there's no class on Friday. So I could make that 10 a.m., but it'll be up Friday morning, and you'll have till Monday morning at class time to get that submitted. And I'm having trouble with the Canvas grading feature, whether it lets you know your answers, or I'll have to figure out how to do that. And because there will be some short answer questions, I have to figure out how to make all that work for the exam. So I have some things to figure out. And I'm going to, for the 3+1 students, ask about a help session. So I will have that, and you're all are welcome. It'll be the same Zoom account. So that'll be Wednesday night at 8:30. Okay, and you have to come with questions because I'm not going to have any kind of formal material ready to present. Okay, all right. Real quick quiz review. Looks like most of you did really, really well. There's a question about, oh, that was a fun question. I think, let's see. You had to figure out a D value just based on some language here. Same kind of thing that I gave you in class. Heat penetration affect thermal processing. We talked about that, the cold spot. We talked about deriving D values empirically, not by guess. I showed you this graph and you had to figure out the Z value. You got to tell me the name of the graph. Here, the pH question, which is the threshold, acidulants, and the issue with herbs and spices as antimicrobial agents. Okay. All right, so now we're caught up and we're going to move on to our next category of antimicrobial strategies. And we're still talking about killing microbes. So we talked about thermal. We spent a lot of time on thermal kinetics, and then we wrapped that up by talking about not really new technologies, but aseptic packaging and the flexible retort pouches. And then we went through a rather long, I always talked longer on irradiation than I intended to, because it just doesn't have very many applications despite its effectiveness. Okay, so now we move on to other new generation processes, and some of these have been widely adopted, some are emerging. Okay, but the rationale for these types of technologies is due to that clean labeling that we talked about before, minimally processed, non-thermal, generally non-thermal, to inactivate microbes. And sometimes you'll see this as, I don't like this term very much, but invisible processing, because you really can't tell that the food has been processed. So I suppose, if you had corn in a can or a vegetable in a can, you could tell that wasn't fresh, even if you didn't know that, even if you never saw the can, you could tell it's been cooked more. and more processed. So these are things that are intended to be like fresh broccoli, okay? And I guess that irradiation could fall into that category. But really we're talking about some of these newer technologies, HPP, high pressure processing, PEF, pulse electric field, ohmic heating, which does have some heating to it, high intensity light, and a kind of a rather new one called cold plasma technology. Okay, so we'll start with HPP, and there's a HPP unit. I don't know if any of you work in the pilot plant or have worked in the pilot plant, but there is a HPP unit in our pilot plant. It's a pretty good size unit. And I think there's even two of them. There might be a small one and then a pretty good size one. Okay, what's the principle of high pressure processing? So high pressure kills microbes. Okay, and this just shows you the range of pressure on planet Earth. So from way up in the mountains, which is low pressure, all the way down to the bottom of the oceans, which is high pressure. Okay, and it's down these high pressures that will kill microbes. Okay, and this is a paper from 1971 in Science magazine. It's kind of cool. What they report in this paper was that so in Woods Hole, Massachusetts, there's a oceanographic institute. It's a great place. I've been there before. And they send out these little, some of the researchers go out in these little kind of submarines. They're submarines. They go out there and they explore. And one of these submarines had a accident. And so the people in the submarine had to be rescued, which they were. But they had to leave their lunch behind. And when they came back, many months later, the sandwiches were okay. They could have been eaten, okay? And it's because under that high pressure, microbes couldn't grow and couldn't spoil that food. So that was a kind of a cool discovery. And so now it's applied as HPP processing or Pascalization, okay? So very high hydrostatic pressures, 40,000 PSI, 300 megapascal for a period of time. You can disrupt non-covalent bond, denature proteins and macromolecules, and especially cause damage to membranes, bacterial membranes, while providing few adverse effects on foods. So this technology has now been widely applied to a whole range of foods, jams, fruits, and, other fruit products, applesauce, salsa, meat and seafood, but especially hummus, especially guacamole. So, and I'm going to talk more about guacamole in a second, but well, actually, I'll just mention it now. So, if you like guacamole and try to make it at home, you would know that you cut the avocado, but you don't want to make that guacamole too far ahead of time, because once it's exposed, it's subject to enzymatic browning, and it will brown pretty quick. So you have to do something to prevent that, keep the air out, acidify it. Those are two good ways, but it will brown pretty quick. And then the shelf life of guacamole is also not very high because of microbes. So avocados do not have a very low pH. I think it's near neutral. There's sugars in there, there's fats in there, so it will not last very long. Certainly, if you make guacamole and try to sell it in a grocery store, you have to do something to it to keep it preserved. And back when I was a college student, in fact, until the 1980s or so, it was hard to find, guacamole was available at restaurants. They made it kind of on the spot, but you didn't see it at grocery stores because it was too hard to preserve for weeks and weeks. So it was due to HPP technology that guacamole became now a staple. So you can go anywhere and get all sorts of brands. It's very shelf stable, quite good, and it's almost all processed by HPP. And I'm going to see if my link can work here. So hold on a sec. So give me a second, I might have to back out of the stop share for a second. Okay, so. Sorry, I have to go through all these manipulations. So this is HPP for a
At Sandridge Food
meat
Corporation, we're
application.
proud of the high-quality, fresh-tasting products we provide customers. We won't tell you all the secrets to producing such great products. What chef would? But we can tell you about one innovation that enables us to provide food that doesn't need added preservatives and yet stays safe and clean. That's our HPP machine. HPP stands for high pressure processing. By using cold water and extreme pressure, we're able to kill all the harmful bacteria in our foods. And it's an amazing process. Let's walk through how it works. And it starts here with food worth respecting. First, finished product that has already been prepared in a clean environment using food safety processes in our facility is transferred onto the HPP's conveyor belt. From there, the product is loaded into cylinder carriers. 4 of these carriers are filled before moving forward to the inner high-pressure chamber. Once all 4 carriers are in place, the entire high-pressure chamber moves toward the center of the machine, aligning itself with the water pipes. Next, the high-pressure chamber is filled with low-pressure water to evacuate all air from the chamber. Once the air is gone, the chamber is filled with highly pressurized water. The HPP machine will exert up to 87,000 psi, equally on all sides of the product, for up to 3 minutes. This special process works on a cellular level, destroying the cell walls of harmful bacteria while maintaining the food's original flavor and consistency. What an achievement! We don't need to add preservatives to achieve extended shelf life, and the true flavors of our ingredients shine through, which creates a better tasting product. Finally, when the high-pressure process is complete, the pressure is released, the water emptied, and the chamber with the 4 carriers moves back out toward the roller track. Now the next 4 carriers are moved inside the high-pressure chamber, transferring the processed product toward the unloading side of the machine. The bacteria-free product is then transferred to another conveyor belt, where it goes off to be dried and packaged for shipment. The HPP machine is an exciting example of how Sandridge Food Corporation lives up to our motto, where culinary and industry come together. And the proof is in our premium.
So that was so I think that was a company that does contract HPP manufacturers, what they do. Okay, have to stop share again. Okay, so just a little video to show you the process. But here's note, it's not continuous, so they do that batch at a time. So it does. In food industry, we love things that are continuous. So that's not continuous, and so that's the problem. But it still works great. And you can see all sorts of products, meat products, salad products, salsa, hummus, pasta sauce, but especially those guacamole products. So Boley Guacamole is a big brand, and they use HPP. Okay. There's just a few more pictures. This is actually an HPP operation here in Lincoln. Universal has some of these devices, and I think they do contract manufacturing. Okay. All right. The next one we'll briefly talk about is PEF, pulse electric field. Involves pulses of electrocuting foods through tubes, and the electric pulse causes membranes to become permeable and leaky, and then the cells die. And the main application is liquid eggs. And I think it gets used some, but not a lot. Another new one is called high intensity light. It involves pulses of white light in the UV to IR range, infrared range, and is used for surface decontamination. And I'm gonna go back out and just go to another video. Sorry for that. See if I can find this one. A lot of trouble just to show one quick video. So all they were doing there was flashing a light over those containers to inactivate microbes that were in the containers. Okay. Find my slides again. Okay, and then the last, I think the last new technology is called cold plasma. And plasma is the fourth state of matter. So there's liquid, solid, and gas. This is different from plasm, like cytoplasm or blood plasma. And it consists of ionizing gases with high ionized gas that contain high energy electrons and other reactive oxygen species. So we saw these things before, these reactive oxygen species, when we talked about irradiation and radiolysis, and even the oxidation reactions. So these reactive particles kill microbes. It's made either by, well, it's made naturally as lightning, but it could be made by discharging technologies, which I will not go through. And it kills microbes by inactivating membranes like we've seen before, and cell walls and activating enzymes. And the targets can be pathogens or spoilage organisms. And this is getting a lot of attention. It's getting a lot of attention. So in food science and for fresh produce, juices, meats, and surfaces. And has anybody heard of cold plasma more recently? In what context? It's being used as a surface decontamination for coronavirus. So kind of interesting. All right. You can see here's meat going in through this tunnel that is exposing these microbes. And this just shows a little bit of a effect on the cell by breaking cell membranes and so forth. Okay, so that's all we'll have to say about the killing and activating part of our overall strategy. So remember, there was keep out, remove, inhibit, and then part of the inhibit we skipped. We're going to come back to that next. Number 4 was killing, and number 5 was combination. So we're going to go back to inhibiting, and there we talked about inorganic agents, organic agents. I think one was called natural, and now, and sort of related to the natural, they kind of broke it apart, are antimicrobial agents produced by microbes, biologically based. So we kind of skipped this one because I wanted to make sure we had enough. I wanted to cover the killing so that you could do your problem set before we had the exam. Okay, so there are a number of approaches here. One is antibiotics. Now, Can you put antibiotics in foods? Nearly all are illegal. Why are antibiotics? So that'd be really effective. Put tetracycline or chloramphenicol in foods. That will definitely kill microbes. Why is that illegal? Not just not warranted, but illegal. Why is that illegal?
Because that could make like people resistance to antibiotics when they get disease.
Sure. So if you expose microbes to antibiotics a lot, you will select for resistant microbes, and you will select for actually resistant genes. And those resistant genes can find their way to a pathogen. So then when you get sick and have pneumonia or strep throat or something like that, the antibiotic is no longer effective. Okay, but there's another reason. What's the first question you get asked when you go to one of the first questions when you go for a doctor visit?
Besides, there is like a microflora bacteria in the GIT, so probably the antibiotic is going to kill some of these very beneficial microbes.
Okay. Yeah, perhaps, but what's the first question? Again, I'll go back. What's the first question you get asked when you go to a doc? Exactly, Emily. Are you allergic to any antibiotics? That's exactly one of the first couple of questions after your name and your birthday. Okay. So there's a lot of people that are allergic, and then we have all this antibiotic resistance that we're concerned about. Okay, there's one exception. It's an antibiotic called natamycin, also known as pimericin. It's produced by a soil microbe called Streptomyces, and it's an antifungal agent. It has antimycotic activity. And why is it allowed? Because gene transfer among fungi is relatively rare, and it's not used clinically, so we don't worry about the resistance factor. It has just a few applications: surface of cheese, dry sausage, wine corks, perhaps a few others, but it's only when we're targeting the antimicrobial to the particular problem. So where there's a problem with mold, like surface of cheese and sausages, wine corks, that's where this agent gets used. Okay. The other category that we're going to spend several minutes talking about are bacteriocins. Okay, so these are not antibiotics. They're not antibiotics. It's a whole different category, but they are biologically based antimicrobials. So bacteriocins are antimicrobial substances produced by bacteria that inhibit other bacteria. And among the producers are lactic acid bacteria and other food grade bacteria. So there's a whole bunch of microbes that produce bacteriocin. So E. coli produces bacteriocin, Pseudomonas produces bacteriocin, but so do food grade bacteria. Why do microbes produce bacteriocins? Why would they produce substances that inhibit other bacteria?
Because they competed with because of the competition.
It's a competition, exactly. So if they share an ecosystem and they want to manage the competition, they produce agents that inhibit others. They have several shared characteristics. They're usually small peptides, so less than 10,000 molecular weight. They're often heat stable. Some of them can even survive pasteurization or even higher temperatures, higher than the microbe. Their spectrum is usually narrow, so closely related organisms. But, so again, lactic acid bacteria are gram-positive microbes, but there are other gram-positive microbes that are important that we don't mind inhibiting. So many of the gram-positive pathogens are inhibited, like staph, Clostridium, Listeria. And even spoilage organisms, like other lactic acid bacteria, are inhibited. Their primary mode of action, so some of them can disrupt cell wall biosynthesis, but the most common mode of action is disrupt membranes. Okay, it makes the cell leaky, so it leaks. So they become acidified on the inside, ion gradients dissipated, and they leak energy, okay? And so they run out of ATP. But the point is, whether it's cell wall or membranes, it's at the surface. Now they're widely distributed among lactic acid bacteria. So this is a phylogeny of just lactobacillus. Now I don't have, these are the old names of lactobacillus, but you can see, I don't know how many are on here, but a couple hundred lactobacillus species are listed here. And it shows how widely distributed are bacteriocins. And they break these down into the different classes, which I'll get to in a second. But you can see the black circles, you can see lots of black circles for which a bacteriocin has been characterized. You can see all, and you can see the type of bacteriocin by all the circles. So I don't know, it looks like maybe a third of the lactobacillus produce a bacteriocin. Now there's different ways of classifying them. I don't want to get too hung up on, so there's class 1, class 2, and then there's these subclasses based on the primary and secondary structure and tertiary structure of these, and even some of the sequences in these bacteriocins. But what I want to so some of them are cyclic, some of them are dimers, some of them have unique amino acid domains, some are globular. The main point that I wanted to make on this slide is that there are two main classes. Don't worry about 3 and 4. Two main classes. There are those that are unmodified. So the primary sequence of the amino acids is the bacteriocin, okay? Unmodified, and some are called post-translationally modified. So what post-translationally modified means is, remember, DNA goes to make a messenger RNA. That messenger RNA goes to the ribosome and makes a protein sequence, a peptide sequence, okay? If it's unmodified, the sequence that comes off that ribosome is the protein. But the post-translationally modified, it means that after the sequence comes off, the amino acid sequence comes off the ribosome, it's modified after the sequence has already been made. And the best example of that is niacin, and I'll show you in a second what that means. Okay. And sometimes this is called lantibiotics because it contains a unique amino acid. Okay, so here's the mode of action of class 1 and class 2. The best example of a class 1 bacteriocin is called niacin, N-I-S-I-N. This is different from niacin, N-I-C-I-N, the vitamin. Okay, that's different completely. Okay, and here's an example of class 2 lactococcin produced by a lactococcus. This is also produced by a lactococcus, by the way. And you can see they get through the cell wall of the target organism. So they're synthesized, they're secreted, and they're across the cell wall, and then they form pores in the membrane, so things leak out. Okay, that's the mode of action. Okay, pores. So this is the cell membrane of a target, lipid bilayer, but it forms a pore, so things leak out. Okay, so now let's focus on niacin, which has a long history. So almost 100 years was that when it was discovered, approved almost 40 years ago in Europe, and 30 years ago by the FDA as a food additive. Now, to get approval as a food additive takes a long time, and it means that the material, whether it's a antimicrobial, maybe it's some sort of organic acid, or it's a protein, or whatever it happens to be, it has to, when you submit it to get approval as a food additive, it has to be in a very pure form, okay? And I'll come back to that later. But it takes a lot of work to purify it, took a lot of work to get it approved as a food additive, years and years. And we'll come back to the importance of that in a moment. It's produced by different strains of Lactococcus lactis, subspecies lactis. This is an organism common in cheese, other dairy products, but only by certain strains of Lactococcus, okay? And it's a really small peptide, 34 amino acids. That's really small, okay? And there you can see the primary, you can see the structure, each amino acid, lysine, valine, histidine, serine, and so forth. And you can see the genes encoding for it. So there's about 10 genes that are involved. So NICE A is the niacin gene that codes for the primary structure, or yeah, primary structure. NICE B are genes involved in the modification after it comes off the ribosome. Okay. NICE T encodes for the transporter function. So the nicin's made in the cytoplasm of Lactococcus. It has to get to the outside. Okay. Another modification. NICE I is an immunity gene, so encodes for immunity properties. So it has, the cell that makes it has to be immune to it. Okay. And then there's genes involved in the regulation of the synthesis, and here more immunity genes. Okay, so you can see all the different genes. Okay, it's inhibitory to L-mono, CBOT, other lactic acid bacteria, and in as far as the legal status, it's the only bacteriocin approved in pure form as a food additive. Okay, and it's sold as nisaplin by Danisco DuPont. Right? You could go to their website, and you can see nisaplin antimicrobial, heat stable, non-dairy based antimicrobial used in a wide range of foods. Reduce food safety risk, protect shelf life, reduce processing times, we'll get to that in a second, meet consumers' demand for natural, because it's all natural. It's produced by a bacteria that's normally used in cheese. Anti-gram-positive spoilage and pathogens, all types of foods. Okay, now here's what I meant by that positional modification. So here is the primary sequence of niacin. The sequence of these has a few amino acids that are involved in excreting the niacin peptide from the inside of the cell to the outside, and then shows the remaining structure. The modification occurs through dehydration of selected serines and threonine. So here's some serine and some threonine, and it forms these kinds of bridges that end up forming lanthionine, which is kind of an unusual amino acid. You don't see it very often. So niacin actually has these bridges. So here's a sulfhydryl, and then here's a bridge here, and here's a bridge here. So it's kind of a unusual structure. Okay, that's what's meant by post-translational. Now there's other bacteriocins other than niacin. So there are niacin derivatives, which we'll look at in a second. There's pediocins, lactocins, other lactic acid bacteria that produce these things. And they are very similar in structure and mode of action. So again, here's niacin A. You can see these bridges. You can see subtilin, all these other ones, lactocin, and they all have very similar structure to niacin. So niacin is not the only one. It's the only one that's approved in pure form. Okay, and you could, since it's so small, it lends itself to protein engineering. So researchers, since there's only so many amino acids, they've gone and they've switched out every single amino acid for another amino acid. And so then they could see if they can enhance its activity through molecular engineering. So I don't know if you could see this. Let me see if I could do something here. Okay, so if you look at this Petri dish, here's how they do this. These are very common experiments. We've done them in my lab many times. So you take a Petri dish, and let's say your indicator is Listeria. Okay, and you take a 1/10 of a ml or so of Listeria, and you spread it on this plate. Use your hockey stick or a swab and spread that Listeria on there. And if you then incubate it for 24 hours or 48 hours, you don't see colonies because you have so many Listeria, you see a lawn. The whole plate becomes turbid. Okay. Now, if prior to incubating. So you spread that listeria on here and you dig out some wells. You just make a I don't know, like a 1 centimeter well, cut out of that agar in a nice uniform circle, or you can take a filter paper and 1 centimeter or even less, half a centimeter filter paper, and stick it onto that agar. But whether it's a filter paper or a well that you've dug into the agar, you fill it with the bacteriocin. So if it's a well, you put 50 microliters of the bacteriocin, or if it's a filter paper, you spot 10 or 20 microliters of your bacteriocin, like niacin. In this case, it's niacin. Okay, then you incubate it. What you will see, you can kind of see around, this is a good example, you see a zone of inhibition, because as the niacin diffuses out of the well or of the filter paper, it inhibits the listeria that would normally grow right next to that well. So you see a zone, okay? Now if we go down here, after engineering that niacin, look how big the well got. So they genetically engineered the niacin. Maybe they switched out some amino acids and it enhanced its activity. Okay, so that's what was done here. Now, would you expect niacin to inhibit O157? Or would you expect it to inhibit Salmonella or Campylobacter? That's a question I'm asking. And if not, why not? Silence. You would not expect niacin to inhibit Salmonella or E. coli or Campylobacter because, by definition, bacteriocins inhibit closely related microbes. Those are gram-negatives. But if you can, again, use engineering approaches, now this would become GMO, but there's other ways you could change the gene structure, maybe without making it GMO. But if you could, then maybe you could, if you could make niacin inhibitory to those gram-negatives, or other bacteriocins inhibit gram-negatives, you really have increased its value quite a bit. Okay, and again, there's now lots of products that food companies have introduced. So here's anattomycin. I mentioned the nisaplin. Now here's an interesting thing called fermentates, MicroGuard. Fermentates. Okay, so what are these things? Here's Christian Hansen is the other big company, and they make similar kinds of products as well. Okay, so we'll come back, we'll talk about these fermentates in just a moment. So, so we talked about niacin derivatives, and now fermentates. So what a fermentated, so I mentioned, again, I've emphasized this before, that niacin is purified, approved as a food additive, very expensive to do this, and yet there's all those other bacteriocins that are commercialized, but they're not food additives. So how can they get put into foods? They're not food additives, approved as a food additives, and they're not purified. So these companies realized they wanted to take advantage of these bacteriocin properties, but they didn't want to go through all the expense of going through FDA approval and purifying. So what they realized was, so niacin is made by not putting Lactococcus lactis in a fermenter and growing it so that it produces the niacin, and then they go through all this purification, expensive purification. But they realized, well, what if we just grew the microbe in the fermenter and didn't purify it? What if we just then harvested the entire fermentation material, collect it all, which contains the bacteriocin, and it contains organic acids, because lactic acid bacteria produce lactic acid, which is an organic acid. We take that whole material and spray dry it, maybe pasteurize it and spray dry it. And the bacteriocin will survive the heating because they're heat stable. So now that material at the end, that powder, is called on the label, cultured skim milk or cultured corn syrup, whatever was in that medium. If we wanted it to be non-dairy, it could just be cultured corn syrup, whatever we grew the lactococcus, okay? And so this becomes label friendly. So these are retentates, and that's what this is. Patented natural clean label range for shelf life preservation, okay? And you can combine these retentates with niacin, with other things, okay? And so it has the bacteriocin and whatever organic agents, organic acids, the production microbe had produced, okay? So if it's a heterofermentative lactic acid bacteria, it could have produced lactic and acetic. Okay, so that's what a retentate does. And I just want to briefly mention Reuteren. It's produced by Lactobacillus, some strains, Lactobacillus reuteri. It also is inhibitory. It's not a protein, and it hasn't been commercial. It's been tried to be commercialized. I won't say too much more about that. And then there are yeast-produced things similar to bacteriocins that are called killer toxins, and they have some application in the wine industry. Okay, and then just very briefly, other biologically based antimicrobials include bacteriophages. Now, that's kind of interesting. Bacteriophages are viruses that infect and lyse bacteria, and there are phages that infect and lyse every bacterium on the planet. So there are some that infect Salmonella, Staph, and Listeria. You can apply these directly or into packaging, like active packaging. And so there are some products like PhageGuard that kill Listeria, Salmonella, E. coli, but there are live viruses that do that. And so there's a little bit of a scary feature to that. Some people don't want to put viruses, even though it's not a virus that affects humans. It only affects these pathogens. They're very specific. And what the virus does, it attaches to the cell, it penetrates the cell, it takes over the cell and bursts the cell, releasing lots of different viruses. And that's how viruses spread. It infects, injects its DNA, takes over the cell makes more viruses, and they go on to infect more organisms. So we'll talk more about viruses later. But the idea is, why use the virus? Why not just take the enzyme that lyses the bacteria? And so that's called the bacteriophage lysine, and that has some applications in food safety. There's companies selling lysins. Okay, and then ultimately, I just want to finish this as yet another company that sells ingredients. One of my former graduate students works for Carry Foods, and again, all sorts of natural antimicrobial agents, and you can see their data. Opportunities backed by research. 84% people want natural, less processed foods. They don't want sodium nitrate in hot dogs and sausages. They want natural on the menu. So these bacteriocins and fermentates have lots of upside. Okay, and then I just want to emphasize the common mechanisms of how these cells work. This is not exclusively how they work, but this is one among the major mode of action, whether it's organic acids, bacteriocins, HPP, PEF, cold plasma. They all target the cytoplasmic membrane, forming pores, disrupting that membrane. When the membrane's disrupted, the cell loses its integrity and dies. Okay, so that's sort of it for inhibiting and killing microbes, but I do have to comment about the survivors of all of these things that we've been talking about, because this is an important area in food preservation. So all the agents that are intended to inhibit or kill microbes may leave behind some survivors, as good as they may be. Now, the only one that probably doesn't is high temperature thermal processing, but just about everything else leaves behind survivors that are, excuse me, that are become injured, if you will, as a result of all these things that we've talked about: chilling, freezing, drying, low pH, sublethal heat, sublethal irradiation, sublethal HPP, metabolic stresses. So they're not dead, they're not real viable, they're injured. Okay, and this has important consequences. So injured microbes may not grow well on selective mediums. So if you design a process to inactivate salmonella, and then you wanted to see did it work, and you plate the survivors, or you plate on a selective media, they may not grow. Okay. Instead, they're viable but not culturable. VBNC, viable but not culturable. Okay. But they're still there. And even so, any of your methods may not show them. But later in the food, they can become viable, grow, and spoil the food, or worse yet, cause foodborne disease. Okay. So now, in most laboratories, they have developed techniques to enumerate those. And they usually involve either enrichment. That's why you do, those of you in the laboratory, that's why you do enrichment. Okay. Now, when you do enrichment, it's not for enumeration purposes, it's for detection purposes, plus minus. Okay. Or use use non-selective medium. So that's why you do enrichment, is to recover injured organisms. And VBNC are all over the place, not just, so they're for food pathogens, but all sorts of other pathogens, tuberculosis, pneumonia, all sorts of pathogens, but especially we're concerned about food pathogens, and just about everyone can exist in the VBNC state. Okay, and even spoilage organisms. This happens to be beer. You could see in beer stored at room temperature or at ambient temperature, there are total lactobacilli, there are viable lactobacilli, and there are culturable lactobacilli. So these are the ones that you would culture, but you can see there's other ones in there too. So the total is up here at 10 to the 6, but you got none on your plating, okay? But there were 10 to the 6 there, okay. All right, and then there's another phenomenon called the stress response systems. This is related to VBNC, but it's a little bit different. Okay, and this is when sublethal processing elicits a stress response. Okay, and a stress response means that you've exposed a microbe to what you thought was going to inhibit or kill them, and it did kill many of them, but the survivors may have induced a stress response so that it helps them survive an even worse stress that comes later. Okay. So sublethal exposure to heat, cold, low pH, starvation induces a stress response. So for example, Listeria is exposed to a temperature shift of normal temperature, 30 to 48. Now 48, remember, that's why I said, you gotta remember those kind of pasteurization conditions. Pasteurization of milk is 62.8 for 32 minutes. So this is way below the temperature that would kill Listeria, okay? It causes a stress, okay? And when that happens, so it's pretty warm, 48. It's pretty warm, but doesn't necessarily kill them. But the stress induces in Listeria a set of proteins called heat shock proteins that the organism makes quickly. And they protect the cell against a subsequent or more severe temperature challenge. So if Listeria could think, it would say, uh-oh, it's getting warm. I better do something in case it gets even warmer. Okay, and those are stress response proteins. Okay, and what do? They are involved in repairing broken DNA, repairing denatured DNA, repairing denatured proteins. They may help physiological pathways operate better. So here is some data. Again, heat shock at 48, been exposed to condition 64, which is very close to milk pasteurization. Okay, and so what we see, now they're measuring D values. Okay, time to kill 90%. The higher the D, the more resistant. So here's 3 different treatments. We'll just look at the average. So this is the D at 64 after heat shock. But here's the control, no heat shock. The D at 64 is 3.3 minutes. If it was given a 30-minute heat shock, 30 minutes at 48 degrees, the D went up almost a minute. If it was sat there at 48 for 60 minutes, it went up another half a minute. And if it sat for 2 hours, giving that cell plenty of time to induce a stress response, the D value more than doubled. Okay, and then this just shows it here on our survival curve. Okay, control, no heat shock, with heat shock. Survival curve. Okay, same thing happens at pH. Given an acid shock, acid shock in this case is sodium lactate, lactate, acetate, acetate, or no acid shock. And the ones that got the acid shock, well, not all of them, but the one that got the acetate then survived a pH 2 much better. Okay, and I could show you more data, and I'll save some of this for Wednesday, but it happens for almost every pathogen and for almost every kind of treatment. Okay, so we'll stop there. A little bit more on this VBNC on Wednesday, and then we'll talk about the last part of our 5-step Hutkin strategy. Keep out, remove, inhibit, kill, and the last one was combination, which we call the hurdle concept. Okay, so we'll stop there and see you all on Wednesday.